Hi everyone, this is Miss Lazar, and today I'm going to be teaching you about DNA structure and DNA replication. Take a moment, pause the video, read through these incredibly high quality memes. When you're ready, let's talk about the lesson. We're going to answer 3 questions today. Number 1, what is the point of DNA? Number 2, what is the structure of DNA? And number 3, how is DNA replicated? During the lecture, you will be taking detailed handwritten notes. You might be drawing some diagrams. You will write down everything that I put on the screen, all the text, but then you'll also be writing down some of the words and phrases that I say that might not be written on the screen. So take a look at the template that shows you what should be in your notes and make sure you're taking super detailed handwritten notes during the lecture. You can always write down more than what the template is suggesting. So, first question, what is the point of DNA? DNA is like the reference book of how to make you. Every cell in your body has a giant set of DNA strands that are held within the nucleus. So we call each really long strand a chromosome, and altogether you have 46 chromosomes in each of your cells, except for your sex cells, eggs and sperm. But we'll get to that in a future lesson. Just like chapters in a reference book, different sections of your DNA contain different instructions. There's instructions for how to be a nerve cell, instructions for how to be a skin cell, and how to make the pigment melanin that gives your skin its color. Although each cell holds the entire encyclopedia of you, all 46 chromosomes, each cell only reads the sections of the DNA that give it the instructions it needs to do its job. So a heart cell just ignores the instructions about how to be a liver cell. We'll talk about how cells determine what instructions to look at and also how they read and follow those instructions in a later lesson called protein synthesis or transcription and translation. For today, we're just talking about how the structure of DNA allows it to store information, and we're talking about how DNA is replicated so that none of the instructions are lost. So we're on to question 2. What is the structure of DNA? And the first thing to note is the name. DNA stands for deoxyribonucleic acid, and the 3 key letters of deoxyribonucleic acid that make the acronym DNA are underlined. Yes, you need to write down this full term. Yes, you need to be able to pronounce it and spell it later on. DNA stands for deoxyribonucleic acid. Feel free to practice that term out loud right now. Deoxyribonucleic acid. So, DNA is a long string like molecule made of 2 strands connected together by rungs that's twisted into what's called a double helix shape. You're going to see many different representations of DNA today. This is the most accurate one, this twisted ladder or double helix shape. However, this twisted ladder form is actually really hard to read because there's all of those colored bases, which aren't colored in real life, but are helpful when we're trying to think about the structure of DNA. And they're really hard to see and read when it's all twisted into that double helix. And so we need to untwist the DNA to see the details of its structure. By the way, when I say double helix, here's what I mean. You can see a single helix on the left, it's just one coil, and a double helix on the right, which is 2 strands that are coiled together. But most of the time when we're looking at models of DNA, we look at it untwisted in this ladder model. So now DNA kind of looks like a ladder on the right-hand side of your screen. We can see the backbone of the ladder, those gray stripes going up and down, and we can see the rungs of the ladder, the different colored pieces. And you can notice that there's 2 colors that make up each rung. That will be relevant momentarily. So I'm going to be referring to the sides of the ladder and the backbone of the ladder, and you want to understand what I mean by that. The sides of the ladder being these pieces that go up and down, and the rungs of the ladder being these pieces that stretch horizontally. The sides of the ladder, the sides of DNA, are made up of alternating phosphate groups these yellow things, and deoxyribose sugars, these blue pentagons. We're going to talk about each. So the phosphate group, this yellow piece that I'm circling, is made of one phosphorus atom and four oxygen atoms. That's what PO4 means. One phosphorus atom, four oxygen atoms. Deoxyribose sugars, these blue pentagons, are made of five carbon atoms, 10 hydrogen atoms, and four oxygen atoms. You don't need to memorize either of those formulas. This is just to remind you that we can talk about molecular structures that are composed of atoms bonded together. And we'll represent them in different ways. We might represent this phosphate group as a yellow circle, this deoxyribose sugar as a blue pentagon. And you just want to keep in mind that this blue pentagon is made of multiple atoms that are all connected together. And they're connected together by a particular type of bond, a covalent bond. And this is something that you do need to know. The bonds holding the atoms together in each building block, as well as the bonds holding the building blocks together, these phosphate and deoxyribose sugars together, are strong covalent bonds. You can think about these types of bonds as the equivalent of a knot tying two ropes together. They take some work to make, they take some effort and energy to make, and they're pretty strong. If you pulled two ropes that were tied together, you wouldn't be able to easily pull them apart. Now let's look at the rungs. So the rungs, these horizontal pieces that stretch along the sides of the ladder, these are where the information is stored. That might be easy to see because the backbone, these sides of the ladder, are exactly the same. We just see phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar over and over. There's no difference whether we go up and down on the molecule of DNA. So there's no information stored there. The difference is the code are all in this series of rungs. Each rung is a pair of nitrogenous bases. Notice that there are two things stuck together to make up each rung, and you can already start inferring what these nitrogenous bases are made of. There are four different kinds of bases. Unlike the backbone, where there's only one type of sugar and one type of phosphate group, there's four different kinds of bases. Adenine, which we abbreviate A, thymine, which we abbreviate T, guanine, which we abbreviate G, and cytosine, which we abbreviate C. Again, listen to my pronunciation because you'll need to say these terms later. The nitrogenous bases consist of adenine, thymine, guanine, and cytosine. Each nitrogenous base is made of carbon, hydrogen, oxygen, and nitrogen atoms. The nitrogen atoms give them the name nitrogenous base. The nitrogenous bases, notice, are always attached to the deoxyribose sugar. They are never attached to the phosphate group. That will be important. You'll need to understand the structure. And just notice that the nitrogenous bases along both sides of the ladder are always attached to the sugar. Each rung is actually made of two bases paired together, and they're always paired together following base pairing rules. These are the complementary base pairing rules. A always pairs with T, and C always pairs with G. Adenine always pairs with thymine, and cytosine always pairs with guanine. We say that adenine is the complementary base to thymine. Notice these are complementary base pairing rules. The bonds that are holding the atoms together in each base are strong covalent bonds, but the bonds holding the two bases together are weak hydrogen bonds. You can see those noted on this diagram as these pink dots that are stretching between the bases. You can think of these types of bonds like two things that are connected together by magnets. They will be held together, but if you pull on them a little bit, they'll come apart pretty easily. And so you can already start imagining that this side of the DNA molecule and this side of the DNA molecule are just held together by magnets, and you might be able to pull them apart pretty easily. That will become relevant later on in this lecture. So let's look at how we actually assemble one of these DNA molecules. Something that's important to know are that there are some free-floating DNA pieces that are always found in the cell. Again, I mentioned that you're going to see many different representations of DNA in this lecture. Here's another representation of the pieces that make up DNA. These pieces of the DNA, the phosphate groups, the deoxyribose sugars, and the nitrogenous bases, are freely floating in the cell, waiting to be connected together into new parts of DNA. But they're usually not alone. These three parts usually exist connected together into a trio of one phosphate, one deoxyribose sugar, and one nitrogenous base. This is called a nucleotide. For example, we could have a guanine base, which would just be this thing in orange, or we could have a guanine nucleotide, which is this trio of molecules connected together. And one thing to keep in mind, you've heard the terms monomer and polymer before. The nucleotide is the monomer of the DNA polymer. That means the nucleotide is the building block of the larger DNA polymer. So, one last time, quick vocabulary review. On the left, we have a guanine base, and on the right, we have a guanine nucleotide. So, listen carefully to what me and your other teachers say over the next couple of days. When we refer to a guanine nucleotide, we're referring to this whole set. And when we refer to a guanine base, we're just referring to this letter, this smaller molecule right here. Let's build some DNA. We've got these freely floating nucleotides in the cell. Let's assemble them into a DNA molecule. And you can probably infer how we're going to do this. So we'll start by building one side of our DNA first. I have a guanine nucleotide. I'll connect that to a cytosine nucleotide and connect that to an adenine nucleotide. I've just built one side of the DNA molecule. Now I'll build the right side. And I'm going to start at the top. What pairs with that adenine nucleotide? A thymine nucleotide, because A always pairs with T. Adenine always pairs with thymine. What's going to connect to that cytosine nucleotide? A guanine nucleotide. And what will connect to the bottom guanine nucleotide? Another cytosine nucleotide. If you only, we can represent these strands in a couple different ways. We can represent it like this, where you can see all of the detail of the different shapes, that the deoxyribose sugar is a pentagon, that the phosphate group is kind of circular, that the guanine nucleotide is this like big hexagon connected to a pentagon, cytosine is kind of a hexagon shape. But we could also simplify it, and instead we could flatten everything out and get rid of the detail of the shapes and just draw everything like a ladder. This would be the same DNA molecule. Notice we've got ACG on the left, TGC on the right. That's the same as this model that we have here. Or we could simplify it even further and we can say, well, we don't need to even draw the backbone at all. We can just write the letters of the bases as we go down each side. ACG, TGC. So same information as before, just multiple ways of representing the detail of the DNA structure. And if you only see one half of the DNA molecule, you can automatically infer its complementary strand. Just like the order of the letters in a sentence determines its meaning, the order of the bases in the DNA determines what the instructions mean. So if we just see one half of the instructions, we can infer the complementary strand because we always know that A bonds to T, C bonds to G, G bonds to C, etc. Okay, now we're ready to talk about how DNA is replicated. And let's start by talking about why DNA is replicated. You already learned that during S phase of the cell cycle, which is after the cell has already created new mitochondria, new ribosomes, new Golgi bodies, and other organelles during the G1 phase, the cell needs to make a copy of its DNA. One copy of the DNA will go to one of the daughter cells, and the other copy of the DNA will go to the second daughter cell. Both copies need to be identical so that the daughter cells both have the exact same set of instructions. And one last time, remind yourself of what that DNA separation looks like, how the daughter cells end up with their different sets of DNA, At the end of G2 phase, we have our two sets of DNA. We've got our duplicated organelles. Those sets of DNA get separated and sent to opposite sides of the cell. The cell pinches in the middle, and eventually we have two daughter cells that each have their own set of DNA. And so it's really important that those two sets of DNA are identical. And we're going to make those two sets of DNA go from one set to two sets during DNA replication in S phase. So now I'm going to zoom in on that process and show you exactly how one set of DNA is copied into two sets of DNA. Okay, here we have yet another representation of DNA. You can see the sugar phosphate backbone on each side. You can see the bases that are paired in the middle. You can notice that they've been set up so that you can visually see that A always bonds with T. It's like two puzzle pieces fitting together. C always bonds with G. Again, two puzzle pieces fitting together. And now we're going to write down the steps of DNA replication. Step 1. First, the DNA is untwisted and the weak hydrogen bonds between the bases are broken. This is done by an enzyme named helicase. An enzyme, remember, is a protein that can break or build chemical bonds. And we can watch helicase zoom through and split open those two strands of DNA. It's able to do that because remember, the bonds that hold these two strands together are just the weak hydrogen bonds, which are like magnets. You can easily pull them apart, and helicase does that. Step 2. Once the two strands are separated, free nucleotides that are floating around the cell start to move toward the unpaired bases on the exposed strands. Step 3, another enzyme called DNA polymerase will use complementary base pairing rules to attach the free nucleotides as a complementary strand of DNA. I'll back that process up so you can see the text again, and also so that you can rewatch DNA polymerase joining those new bases onto each strand. DNA polymerase will make the covalent bonds that connect each nucleotide together along the sugar phosphate backbone. So, for example, DNA polymerase will attach this C to this T with a covalent bond right here. And the hydrogen bonds that actually connect the bases together, remember, think of magnets. These form on their own, just like two magnets snapping together. This, like the knot being tied between two ropes, needs to be intentionally done by an enzyme, and DNA polymerase will do that. What's interesting to note is that as DNA polymerase is walking down the strand, connecting these bases together, it's not perfect. It actually makes mistakes all the time. And it has some proofreading mechanisms built into it so it can catch those mistakes. And some mistakes are caught, but other mistakes will stay there. And that's called a mutation. I'll show you that process in a moment. So DNA polymerase just finished. This process happens on both strands at the same time, as you saw, and it's repeated for all 46 of the chromosomes, all 46 of the strands of DNA that you have in each cell. And at the end, you have two sets of identical DNA molecules. And one thing to notice about these DNA molecules is what they're composed of. I've highlighted the two strands that are from the original DNA molecule. Now I'm highlighting the two new strands that were built. These are called the complementary strands. So we have the original strands that were just separated or pulled apart, and the new complementary strands that DNA polymerase built out of free nucleotides from the cell. This is called a semi-conservative process, and it means that the new DNA molecules are made of half original strands and half new strands. So finally, we've made our two sets of DNA. They're identical. One of the new sets of DNA molecules will be sent to one daughter cell during mitosis, and the other set will be sent to the other daughter cell. But as I mentioned before, sometimes DNA polymerase makes a mistake. So watch the bottom DNA polymerase. As it goes down the strand, something wrong has happened. Instead of pairing a C to this G, it paired a T to this G. And you can see that those just don't fit together. That is a mismatch. it finishes its replication, and we still have this mismatch right here. And so your cells will actually correct this mistake. And remember, those mismatches get fixed during G2 phase. You have written down in your notes from last week that errors from DNA replication get fixed during G2 phase. This is the error that we're referring to. The problem is that your cells actually don't know how to fix this error. They just see a mismatch and they don't know which is the correct letter to keep and which is the letter that we should get rid of. They don't know if this T is correct or if this G is correct. We can look at the other strand and we know that the G was there originally and it should be paired with a C, but many times the other fix will be made. Instead of keeping the G and adding a C or replacing this T with a C, the T is kept and the G is replaced with an A. Now, if we look at these two strands, there's a difference. They're exactly the same, except for this base pair right here. On this strand, we've got a GC pair, which is the correct or original pair. And on this strand, we have an AT pair, which is a difference. This is actually what a mutation is. This is a change in DNA, and this is how it happens. In this situation, if there is a mutation made on one strand, the daughter cell that inherits that strand, this bottom strand, will get the mutated DNA. Our daughter cells will get slightly different sets of DNA. So that's how a mutation is created and how it ends up being passed on to one set of daughter cells. And that's the information that you need to know about. I would highly recommend rewinding this video to rewatch the process of DNA replication, testing yourself, making sure you know which letters pair together, making sure you understand the structure of the DNA backbone, and rewatching any segments that have information you were confused on. But thanks for watching this recording, and we'll see you in class soon.